Okay, thanks everyone. That was great. And we just have a little bit more time now for some additional discussion before we close today. So we just like to open it up to any additional questions or conversations from these first two sessions. This is a chance to, dig a little bit deeper into any particular topic or go into questions that we weren't able to answer with the two previous panel discussions. And just a reminder for the speakers, moderators, and panelists, if you want to answer, to use the raise your hand function. So I think that one thing that Leah and I were listening to that I think got a lot of attention, but maybe didn't quite have enough time to dig real deep into it, is sort of the importance of the critical windows of susceptibility for exposures, particularly in the context of cancer. And Catherine was mentioning epigenomic marks as sort of one way to get at exposures at earlier times. So if there's anything else related to that, of using different omics to get at exposures at different times or different strategies for that, we were going to maybe open with that question. Andrea.
Kim, can I go back to that? We have been successful to get to that question using DNA methylation, as I was mentioning. I put a very informative about, there are about 25 biomarkers that are data driven. Usually people use elastic net to select methylation sites that are informative and helpful to predict anything you might want to predict, whether it's age, whether it's cardiovascular. There is so the reason why DNA methylation is helpful. I would like to hear about other omics actually. It's because DNA methylation, what I understand that happens with DNA methylation is that when we are exposed to anything that changes our bodies, DNA methylation reacts to it. It's a great way we adapt to the environment. The DNA sequence. We think instead about supposed to, and then we can access it and develop biomarkers. There are lifetime biomarkers that have been developed on smoking. And smoking is easy because most of us remember whether we smoked or not and how long. I mean, with certainly some error, of course. And the same, we had developed a biomarker of lead exposure that goes back 10 years in time. So you can measure lead exposure with DNA methylation going back 10 years. Of course, I mean, might seem impractical to use omics to estimate exposures when we can measure exposures, but the advantage, for instance, in DNA methylation is that if you look at the review I put in the chart, you can use the same array, the same measurement to estimate 30 different exposures. So you can think that down the line, it might be easier to measure DNA methylation rather than using different instruments and different technology to measure 30 or 40 different exposures, perhaps hundreds. So this is what I had on that point that I wanted to share.
Okay, great. Dean?
Yes, I completely agree on the methylation. The challenge is really going to be definition of what cell type or what samples we're really going to use. We're going to have to be careful on that. And that really throws me back to another issue for best practices. We have to think about best practices, but we have to do that in a way that is going to encourage development and not stifle it. And that's that's something that we, in some of these areas, we may not be far enough along to really get best practices. But if we get best definitions or very clear definitions and use those definitions in our communications, then I think we can achieve that goal. The last thing I just want to throw out is exosomes. We haven't worked on exosomes, but this seems like it could be a viable way to get at measures. And I think cell population changes over age are going to be really very important. And I don't know if there's a good way to measure that.
Mm-hmm. Okay, Catherine?
Yeah, I very much agree with Andrea and Dean. I mean, I think DNA methylation seems like it's really promising for several reasons. One is that DNA is so much easier to handle than other types of samples. But I think that when it comes to cell types, I think what we want is to be able to look for those cell types that seem like they mean something. Like if we look at Doug Bell's papers on this HRR, right? You see this region that seems like the methylation profile responds to cigarette smoke. Cigarette smoke goes away. It also, that hypomethylation starts to increase again. So you do have these methylation marks that respond, that we have seen, and they seem like it's across cell types, right? As Doug Bell's paper has shown. But then you see some of them that are like the metastable epialleles that Rob Waterland has been talking about, the imprint control regions that we've been working on. Those ones seem like they're stable, but their methylation profiles are limited to a very short period. It's an important period, but it's a very short period before in the prenatal period. So I think the way we should be thinking about this is more not just in terms of the matrix that we use, but also the timing when each of these methylation marks are going to be useful for estimating exposure.
Okay. And Andrea, did you want to comment any more about the potential promise of the exosomes?
Yeah, I mean, the exosomes are very interesting. We have been doing work on exosomes or extracellular vesicles. And as you know, I mean, in our body, as Dean was mentioning, there are hundreds of tissues and thousands of cell types. And when we measure omics, usually we only focus on one or a mix of some. So exosomes are small vesicles that are released by all of our tissues, and they are all coming into the blood or in other type of biofluids. So if you use plasma or serum, you can collect vesicles coming from all tissues. And now in some labs, including mine, we are isolating vesicles that come from specific cell types. So for instance, we can isolate vesicles that come from neurons, that come from oligodendrocytes, so that they come from brain cells, from liver cells. And we have a grand plan now to set up an atlas with at least 25 different cell types, meaning vesicles that come from different cell types. This is particularly interesting because in a way we can use this as a liquid biopsy, and potentially we can measure anything that is in those vesicles. It can be microRNA, we can isolate DNA, we can measure metabolites, and so on. So I'm very happy to, anyone who's interested to hear more, please reach out to me. I'm very happy to collaborate.
Thanks. So one question that I have, it built a little bit on the comment from Dean Jones about best practices. And in that methods discussion and computational methods discussion, we heard a lot of different methods and approaches, and maybe it is like Pete Kraft said, it depends on the question, the methods you're using. But I guess I had a question is like, what are the needs in the field for sort of standards or best practices as we analyze these data with our different methods and computational approaches? I don't know if anyone has any thoughts. Jim, I don't know if you're still on, or Mariana, I saw you pop up.
Yeah, I'm on. That's a pretty big question, Leah. So yeah, I don't know if we can say here's the recipe for best practices for analysis. I mean, I think Pete's right. It really depends on the hypothesis you're looking at, the data that you've got. And it's very different if you have complete lifetime history and you're trying to figure out a longitudinal model with really solid data compared to a situation where you've got little bits and pieces of omic data at different time frames, and you have to focus a lot of energy on trying to build models that you can then use to impute back to your larger cohort. And both are important goals, but they take different thinking and skill sets and methods and things like this. So I hesitate to say there's a one size fits all. There just isn't. I think probably what we found is having a lot of people with varying levels of expertise at the same table talking about some of these things, because it's gone well beyond the realm of what any one statistician can sit down in an afternoon and just bang out some linear regressions or something. You really need people at the table who can provide input into how these models should be constructed, what kinds of exposure response relationships we might expect, linear, nonlinear, what kinds of additional data can be brought to bear. And that just takes, several people sitting at the same table. So to us, that's been really important.
Thanks. Mariana, you were going to say something.
Sure. Yeah, I was also, well, I agree with everything that Jim said. Absolutely, having different stakeholders, whether it's people who are generating the data, clinical experts who have a better sense of the clinical questions and outcomes to link with, is super important. But I will also say it's important to figure out how to integrate. Are there standards for data generation? For instance, for non-targeted analysis, we've done a lot of analysis, again, in other contexts, non-cancer contexts, but I've not integrated mass spec data across studies. So I have no idea how difficult it is. We've figured out how to do that fairly well for transcriptomics and genetics. We're figuring it out for single cell transcriptomic measures. I think there are a number of methods for that, but other types of data are much more challenging, like the microbiome, for instance. We've done some meta-analyses in that space, and it's very hard, even within 16S, to get the data on the same level to be able to aggregate and integrate. And I think that's something to consider, to figure out what are the kind of standards in terms of data generation, in terms of sharing information to enable some of these integrative studies, and how to do that effectively. And that's just one example, but also some of the epidemiological data sets of exposures are the same techniques being used to measure pollution variables and water contaminants and other things across different states. So can we integrate data from the Callan virus screen in California with similar data sets in other states and all over the world? So anyway, it's hard.
Thanks. Marilyn, you have a hand up to see.
Yeah, I was just going to add, I mean, I think this point about best practices and standards is critical. And on the environmental variables, and I guess I'm more so thinking when you are going to capture them through participant reported instruments, more so than leveraging public data sources. I just thought I'd bring up that there is a resource funded by NIH, actually NHGRI, called the Phoenix Toolkit. And that resource has been around for 10 or 15 years now, and they have been aggregating different instruments in lots of different areas of particular disease traits and phenotypes and specific kind of questions and measures that you might have that are disease specific, but then they also have some that are more broad. They have some that are, you know, demographic type variables. They also have social determinant variables. But I think what they've done really well is to pull together experts. So they put together expert teams who review different instruments and measures and come up with standard sets and standard harmonized instruments and variables. And I think especially as we start to think about, you know, some of the earlier comments about we really need broad, you know, really large samples where everything's been collected in the same way so that we could start to aggregate data or have these really large studies. I think looking at those measures, because a lot of those have come from big epidemiologic cohorts. They've looked at how those instruments have been measured previously and used previously, and they're using them or putting them out for others, rather than come up with yet another set of instruments and another set of standards. So looking to the experts in the field for how they've done it, and then using those if we're going to capture new sets of variables on individuals in large studies, just so that we don't have now two sets of standards that we can't combine.
It looks like Pete has his hand up.
Yeah, no, I agree. If you're starting a new study, new data collection, you should definitely leverage sort of what's been out there. And Phoenix is a good place to start to see what's available. But just sort of riffing on a comment in the chat from Tom about harmonization across different, I think that's metabolomics platforms, right? So it's you can't go back in time and you know, regenerate the data using a new platform, and in 2 years it'll be different anyway. So I think given existing data where we have questionnaires or previous biomarker measurements, you know, we're going to have to think about how do we make it easier to combine those data sets. So I think that goes to what I was trying to get at in my earlier comments, which is, you know, at a minimum, documenting how you've generated these data in a way that makes it feasible for other folks to see how does that play well with the data that I've collected. So for some of the questionnaires, you know, just making sure you're sort of mapping things to the same concept ID, and then you have a detailed description of like really what was the question, and then people can figure out how they can map that across different studies. Or again, if they even want to, it's sort of like this is too broad scale. It's not giving me the information I really need to be able to do the study, so that you can really hone in on the data sets that have the information you need. So better is good, but let's not make the perfect the enemy of the good.
Thanks.
There was one other question that I think, Peter, you answered in the chat, but I was wondering about the PRS question. I think the question was if it ever reflects past exposure, or, you know, what the particular uses of PRS might be for exposure, kind of a circuit, basically.
So I'll jump in since I did answer that in the Q&A. You know, if you think of it as a proxy for an intrinsic exposure, so it could be like if you have I'm making this up, if you had a PRS for circulating estrogen levels that you trained in premenopausal women, say adult premenopausal women, you could say that PRS sort of reflects the, you know, average or integrated dose of exposure to estrogen over time. So that people who have the higher PRS will tend to have a higher lifetime exposure to estrogen, and people who have a lower PRS will have a lower estimate. So it's not that it is actually measuring, it's not a response to the exposure, right? Unlike, I think, methylation, the idea is you smoke, these marks change, so we can use those as a proxy for your smoking history, even though we haven't asked you about your smoking history. So the PRS is, I think, a little different, but it also brings challenges. And I think, you know, Genevieve alluded to this, is that there's also a matter of when exposure windows count. the PRS on some level is not necessarily restricted to those exposure windows. It's reflecting potentially exposure across the entire life course, or maybe it really is only capturing adult estrogen levels and has nothing to say about pre-adolescent estrogen levels. So those are, I mean, again, I think the timing would be something to really think about when you're looking to use the PRS as a proxy for exposure, given that the PRS itself is not time varying.
Thanks. We're looking through some of the questions. There are a lot of points in the chat of potential resources, and the NIEHS environmental health language, which is in the chat. There's some comments about ontologies in the chat. Just want to point people to that. But Kim, I don't see another sort of burning question. Do you?
No, does anybody else have anything else they'd like to bring up? Anything that didn't get brought up or that they feel like is a important point that we didn't really touch on with these two session topics? Maybe we're all talked out for today.
All right, should we, if we have no more questions, should we close? All right, I want to just thank everyone today for your participation. Thank you to our speakers, moderators, and panelists. We had really a great discussion. We're going to start again tomorrow at 11 a.m. Eastern time. We'll be talking more about the specifics of environmental data. We'll be talking about model systems. And we'll end with a final discussion where we'll talk about the themes across the past 2 days. So thank you everyone for joining us today, and I hope you have a great rest of your day. Thank you.
Thank you, everyone.
Thank you. See you tomorrow.
Bye.
All right. Bye-bye.